Assalamu alaikum. My name is Muhammad Ali Haider, and today we'll be discussing the pharmacology lecture number 14, and this will be the signaling mechanism and drug actions. Until now, we have considered receptors, interactions, and drug effects in terms of equations and concentration effect curves. We must also understand the molecular mechanism by which a drug acts. Such understanding allows us to ask basic equations with important clinical implications. Why do some drugs produce effects that persist for minutes, hours, or even days after the drug is no longer present? Why do responses to other drugs diminish rapidly with prolonged or separated administration? How do cellular mechanisms for amplifying external chemical signals explain the phenomena of spared receptors? Why do chemically similar drugs often exhibit extraordinary selectivity in their actions? And do these mechanisms provide targets for developing new drugs? Most transmembrane signaling is accomplished by a small number of different molecular mechanisms. For each type of mechanism has been adopted through the evolution of distinctive protein families to transduce many different signals. These protein families include receptors on the cell surface and within the cell, as well as enzymes and other components that generate, amplify, coordinate, and terminate photoreceptor signals by chemical second messengers in the cytoplasm. This section first discusses the mechanism of carrying chemical information across the plasma membranes and then outlines key features of cytoplasmic second messengers. Five basic mechanisms of transmembrane signaling are well understood. Each use a different strategy to circumvent the barrier posed by the lipid bilayer of the plasma membrane. The strategies use a lipid-soluble ligand that crosses the membrane and act on intracellular receptor. Number second, a transmembrane receptor protein that intracellular enzymatic activity is elastically regulated by a ligand that binds to a site on the protein extracellular domain. A transmembrane receptors that bind and stimulate a protein tyrosine kinase, a ligand generated membrane ion channel that can be induced to open or close by the binding of a ligand, and transmembrane receptors protein that stimulates a GTP binding transduces protein G protein, which in turn modulate production of intracellular second messenger. Although the five established mechanisms do not account for all the chemical signals conveyed across cell membranes, and they do transduce many of the most important signals exploited in pharmacotherapy. Intracellular receptors for lipid soluble agents. Several biological ligands are sufficiently lipid soluble to cross the plasma membrane and act on intracellular receptors. One class of such ligands includes steroids, corticosteroids, mineral corticosteroids, sex steroids, vitamin D, and thyroid hormone, whose receptors stimulate the transmission of genes by binding the specific DNA sequence near the genes whose expression is regulated. Many of the target DNA sequence called receptor response elements have been identified. The gene active receptors belong to a protein family that evolved from a common precursor. So we will be discussing more about this pharmacology in our lecture number 15. Stay tuned. Thank you very much. Take care. Allah Hafiz.